This is my oral presentation for the enzyme lab. So a little bit about enzymes before we get started. An enzyme is a biological catalyst that aids in chemical reactions. And enzymes work by binding to the active site of a substrate. And once they bind to this active site, it leads to conformational changes in the substrate and then in fact lowers the activation energy. And enzymes are really important for living organisms because they speed up and regulate chemical reactions in living organisms. And that's important because it allows cells to acclimate to different changes in their environment, which in fact keeps the cells and the organisms alive. So the specific enzyme that we were working with in this experiment was trypsin. And trypsin is an example of a protease, and that is a type of enzyme that breaks down proteins into peptide bonds. So trypsin is found in the small intestines in humans and also in bacteria. It is also secreted by bacteria. So when it's found in the small intestine, it is most optimal at 37 degrees Celsius and then a pH of 8. And when it's secreted by bacteria, the most optimal range for activity varies. Salinity, pH, and temperature are all variable. So our question today was, what does the effect of temperature have on trypsin? And we hypothesized that if temperatures were to increase above 47 degrees Celsius, that trypsin activity would decrease. And our rationale was that in the first experiment, we believe that 37 degrees Celsius would be most optimal for trypsin due to it being most optimal in the small intestine at 37 degrees Celsius. But we soon found out that that was not the case and that 47 degrees was actually much more optimal. And we learned that just because the enzyme is most optimal at 37 in the small intestine does not mean it would be the same in an environment such as a centrifuge tube in our lab. So we had to adjust accordingly. So about how we went about our experiment. So we had 24 total centrifuge tubes, and all 24 of the centrifuge tubes received 450 microliters of reaction buffer. And the reaction buffer had a 0.15 molarity of sodium chloride and a pH of 8. And it was important for us to have a reaction buffer that was the same amongst all of our centrifuge tubes, because if we were to have different molarity or different pH, or different salinity, in this case, different salinity and different pH, that would be another variable in our experiment, and we wouldn't actually be testing for temperature. And we also added 500 microliters of our substrate azocasein, and azocasein is red and orange in color, which is very important later on in our lab. And then 20 out of 24 of our centrifuge tubes received 500 microliters of the enzyme trypsin. And our negative controls did not receive the trypsin, and that was because having the negative controls allows us to see how much activity is actually happening in the presence of the enzyme in comparison to without an enzyme. So we had 5 different temperatures. We had 37, 47, 50, 60, and 70 degrees Celsius, and within each temperature we had 4 replicates. So all the centrifuge tubes were put into water baths at their desired temperatures and were put there for 20 minutes. And after they were done their 20 minutes, they were taken out and we added 500 microliters of 10% TCA. And TCA was important in this enzyme or in this experiment because it stopped the reaction. So before we put our centrifuge tubes into the centrifuge, we wanted to stop the reaction. That way, after the tubes were put into the centrifuge tube, we'd be able to have a separation of pellet and supernatant. So the pellet in this experiment represented the undigested protein, and the supernatant was the liquid at the top that was either a light yellow or a bright orange, and that represented the digested azocasein. So after we separated the supernatant from the pellet, we then used a spectrophotometer and read the absorbance reading at 400 nanometers, and that was important because azocasein has sulfonamide groups, which these groups absorb light at 440 nanometers. Okay. So as for our results, here are all the different results we got from each reading. And here's a bar graph representing the trypsin enzyme activity versus temperature. So down here we have 37, 47, 50, 60, and 70, and different absorbance readings. And you can see that 47 degrees Celsius had the highest bar which shows us that it has the highest enzyme activity. However, 50 is also pretty close up there, and their standard deviations have overlapping in the graph, so that shows us that they're not significantly different. In fact, from the p-value that we received from the 47 degrees Celsius and the 50 degrees Celsius showed that they're not significantly different because the p-value is greater than 0.05. However, our standard deviation amongst all trials was low, was only 3 to 6%, and that shows that we do not have a lot of variability in our experiment, which is good. In fact, for our 47 degrees Celsius, we only have 5% error or 5% standard deviation, which is low variability. We also have a 3.5-fold increase in enzyme activity when comparing 70 degrees Celsius to 47 degrees Celsius, and that just shows that 47 degrees Celsius is much more optimal than 70 degrees Celsius. Also looking at our p-values, when compared to 47 degrees Celsius, 37, 60, and 70 were all significantly different because all the p-values were less than 0.05. Okay, so our experimental design, I believe, is very adequate. From our first experiment, we did learn how to make the experiment run more smoothly and how to make sure that we're getting the most accurate results that we can. Our hypothesis, we did support, like I said before, 47 degrees did have the highest absorbance reading with an average of 0.53. And sources of variability, so adding the TCA before going into the centrifuge tube is a very important aspect of this experiment. We only had 2 group members administering the TCA, and like I said before, we had 24 centrifuge tubes, which that is a lot, so some tubes were getting the TCA before others, which that could lead to variability. Potential improvements, more replicates is always okay. Having more replicates gives you more room for error. Say if a centrifuge tube was to fall, or you had an outlier, and it also adds more significance to your data. And also, we could have had more members administering TCA just to make sure that all the centrifuge tubes are receiving it at almost the same time to reduce variability. So in relevance to growing bacteria, like I said before, just because trypsin is most optimal at 47 degrees in an environment like this does not mean it will be optimal in a different environment. So for bacteria that are living in a similar environment as centrifuge tubes in our lab classroom, 47 degrees Celsius would be most optimal for trypsin activity. And here are my sources.